Hi, everyone. Good morning. We're going to get started. Today, I have the pleasure of introducing Dr. Andrew Stewart. Dr. Stewart is a physician scientist. He received his Bachelor of Science degree from Trinity College and his medical degree from Columbia University College of Medicine. He serves as professor at Yale, then chief of endocrinology at the University of Pittsburgh, and since 2012 has served as a director of the Diabetes, Obesity, Metabolism Institute at the Icahn School of Medicine at Mount Sinai. His research focuses on human insulin-producing beta regeneration for diabetes. His research has culminated in the discovery of the DYR-K1A inhibitor class of beta cell regeneration drugs, the understanding of the genomic pathways underlying beta cell expansion and insulin overproduction in human insulinomas, and the combined use of the DYR-K1A inhibitor, harmine, and GLP-1 receptor agonist in increasing human beta cell proliferation and reversing diabetes. In 2023, human clinical trials commenced. Please join me in giving Dr. Stewart a warm welcome.
All right, thanks. So very fun to be here. I'm going to tell you a story that's very much a Mount Sinai story. It all began here. It continues here. We continue to be leading this at Mount Sinai. So let me just see if people can hear the pointer works. All right, so here's the deal. First of all, Mount Sinai owns patents on much of the stuff that I'm about to tell you about, so I do have that conflict. Just to set the stage, right now there are 38 million people in the United States with diabetes. There's another 100 million with metabolic syndrome or prediabetes or impaired fasting glucose. There's another 500 million people in the world that have diabetes. It's the second or third cause of death in the US if you include its contribution to heart disease, cardiovascular disease, stroke, and cancer. It's the #1 cause of blindness in the US, kidney failure, limb amputation, liver failure. It accounts for 30% of people in a hospital at any moment. Like right now, 30% of the people at Mount Sinai are here because they have some complication of diabetes, typically. It also accounts for about 1/3 of hospital expenses. The cost in the US, and this is as of 2012, those are the latest numbers, is $400 billion a year. And obviously, its cost to the people who have it and their families is unmeasurable. So it's a big problem. What I'm going to tell you is that at the end of the day, in contrast to what you may have heard over the years, ultimately all diabetes is caused by inadequate numbers of pancreatic beta cells. So let's just talk a little bit about the beta cell. We all have a pancreas. Pancreas is about the size of a strip steak. Scattered through it are 1 million separate individual little islets, little islands of Langerhans. They comprise 1% of the pancreas. And if you add them all up together, all the islets in your pancreas right now are about 1 cc, and they're scattered throughout the pancreas. Their job, obviously, is to sense glucose and secrete insulin. There are a bunch of different cells in the islets. The red ones here are beta cells, but beta cells are the cells that sense glucose and secrete insulin. And they know what your glucose is instantaneously. And if your blood sugar is 78, which it probably is right now if you're young and healthy, if it goes up to 82 or down to 74, they can tell the difference and they turn on and turn off insulin appropriately. So this slide makes the point that in people with type 1 diabetes, beta cell mass is reduced. That's not new news. We've known that for about 100 years. The reason you can publish papers like this in this century is that everybody thought that after decades of type 1 diabetes, this number was 0. In fact, nobody dies these days of type 1 diabetes, so we don't actually know what new onset beta cell mass is in people with new onset diabetes. So these are autopsy studies from people who've had type 1 diabetes for 20 years, 30 years, sometimes 50 years. The point here, though, is that even if you've had type 1 diabetes and autoimmunity for 50 years, in some cases 80 years, there's still some beta cells left. So there's still substrate for regeneration. This is normal beta cell mass. So that was type 1 diabetes. This is type 2 diabetes. And this slide makes the point that type 2 diabetes has a major contribution from beta cell failure. If I'm obese and non-diabetic, my beta cell mass is this. If I have metabolic syndrome, impaired fasting glucose, my beta cell mass is lower. These, again, are all autopsy studies. We can't measure beta cell mass in living people. And if I'm a type person with type 2 diabetes, matched for BMI, matched for sex, matched for age, beta cell mass is lower. So clearly type 2 diabetes is associated with insulin resistance. That was actually the source of a Nobel Prize to Burson and Yallo, who were at Mount Sinai for work done in the 60s and awarded in the 70s. So insulin resistance is absolutely part of type 2 diabetes, but if you have enough beta cells, you can overcome that and secrete more insulin. Okay, so beta cell mass is reduced in 500 million people around the world. Everybody has residual beta cells. So what do? Well, we do pancreas transplants occasionally in people with type 1 diabetes. Typically, we do it with a kidney as well, so simultaneous pancreas kidney transplants. In the US, there are about 1,000 of these per year, so that's fantastic. But we're not going to get to treating everybody with diabetes through these kinds of mechanisms. We also have the option for pancreatic islet transplants. The idea here is you get a pancreas, you isolate the islets, you stick a catheter between the ribs and into the portal system and squirt in or infuse these islets. And this is called pancreatic islet transplantation. It's pretty neat. It's not actually approved by the FDA for a variety of political reasons. But despite its efficacy and its minimally invasiveness compared to pancreas transplant, it's not done that often for regulatory reasons, but also because it's expensive to make the islets and there aren't enough organ donors for this or for pancreas transplant. So that's fueled excitement about taking human embryonic stem cells and reprogramming them and turning them into adult-like stem cell-derived beta cells. So this is fantastic. It's promising. It's moving along rapidly. As of now, in the trials being done by Vertex, they've enrolled so far. This is all sort of behind the scenes, so you don't really know the real numbers, but it seems that they've enrolled about 17 people so far and are following them. My take on this is it's an amazing scientific approach. The progress is amazing. It's also super interesting, but it's hard to see how this gets expanded to 500 million people. So our thought was, look, if everybody with diabetes still has beta cells, why don't we come up with a way to make them regenerate? So what do we know about beta cell proliferation? Beta cells do proliferate during the first 6 months of life. So on the y-axis here is beta cell proliferation. On the x-axis, it's age, so embryonic life, neonatal, et cetera. So the beta cells that we all have right now, we made when we were 6 months old. We made a few more when we were toddlers. But by the time we were teenagers, beta cell replication stops. And in adults, which is the source of the islets we have to study for the most part, there is no replication. So it's also been known for a long time. You simply can't force human beta cells to replicate. That's why people have focused on alternate replacement strategies. So we had done a lot of work before I came here and while I was in Pittsburgh, showing that in fact, if you use gene therapy approaches and manipulate cyclins and CDKs, you can force human beta cells to replicate. But we're not going to use gene therapy in people with kids with type 1 diabetes. So we decided to do a high throughput screen, and we developed a way to do it. Unfortunately, when I was in Pittsburgh, we didn't have the money or the robots to do it. So one of the reasons I moved here was the opportunity to do robotic high throughput screens and have funding to support them. So this is very much a Sinai story. This happened in Annenberg 18. So Peng Wang, with whom I've worked for years, and I did this high throughput screen, and we identified a small molecule called harmine, which is shown here. It inhibits a kinase called DYRK1A, and when in doing that, induces human beta cells to proliferate. So we screened 100,000 molecules. Out of 100,000 molecules, one of them actually worked. So what is harmine? It looks like this. In medicinal chemistry parlance, it's a small molecule. That's code for, it's the same size as aspirin or Tylenol. You can take it by mouth. So this can be a pill. Its target is an enzyme called DYRK1A, and it works by sitting into the pocket that ATP sits into. This is a kinase. DYRK1A is a kinase. It uses ATP to phosphorylate substrates. So harmine gets into that same pocket and blocks it. So it's a DYRK1A inhibitor. So if you look at what harmine does on the scale that I just showed you a moment ago, it gets beta cell replication up to around 2%, 3% or so. Pretty good. On the other hand, if my beta cell tank is 95% empty, it's going to take a long time to fill it up at this rate. So we said, let's see if we can come up with other ways to make it work faster. Can we get higher rates of beta cell replication? Can we be more specific for beta cells? So we decided to look at other drugs that already are in use that are beta cell targeting drugs. So sulfonylureas, meglitinides, GLP-1 receptor agonists. And I had a wonderful graduate student named Courtney Akefi who worked with Peng and I screening these other beta cell specific currently used therapeutic molecules. And the title of this says it all. What Courtney found that is if you take any GLP-1 receptor agonist and add it to any DYRK1A inhibitor, of which there are now many, you enhance beta cell regeneration and they work better. So what are GLP-1 receptor inhibitors, agonists? I don't need to tell you, they're all listed here. Right now, there are probably 60 million people in the world taking them. They augment insulin secretion, they're incretins, that's why they're used, and they're appetite suppressants. But none of them make beta cells proliferate. So the experiment that Courtney did, and this is in the Atrium building down the street, was this. Let's get some human cadaveric islets. We get them from the National Institutes of Health. They provide them, they sell them to us for experiments, and we'll measure beta cell proliferation. So these are beta cells that are staining for insulin. These are beta cells that are proliferating, stained for a marker called Ki67. So Courtney got 20 different sets of islets, treated them with vehicle, nothing happened. Treated them with GLP-1, nothing happened, as expected. Treated them with harmine. All of them have a nice response to harmine, but it's very variable. And then when Courtney combined harmine and GLP-1 together, now she got much higher rates of beta cell replication. So she also showed that this is true not just for GLP-1 itself, but also for Exendin-4, that was formerly Lilly's Byetta, and liraglutide, and lixisenatide, now semaglutide. So none of these drive human beta cell replication, but harmine does. And if you add harmine together with these guys, now you get much higher rates of beta cell replication. So there's a synergy between harmine and GLP-1 receptor agonists that enhances human beta cell proliferation. If you take the data that I just showed you a moment ago and map them on what we did when we were 6 months old, what we're doing now, and what we did with harmine alone, you can see this number is around 7 or 8%. It's off the scale here. But now we have really remarkable rates of human beta cell replication. Okay, so I showed you that we've got beta cell replication, but really you could say, Andy, you showed me pretty red dots and some green cells, but you didn't show me you got more beta cells. So I want to see that you have more beta cells. We also wanted to see that we have more beta cells. So we developed a system together with Sarah Stanley and Adolfo Garcia Ocaña, all here at Sinai. So this is what human islets look like. We get them again from NIH, or there are a couple of commercial suppliers. In a Petri dish, they'll last for a couple of days, but then they fall apart, they stop making insulin, and they're not healthy. So we realized that you can actually transplant them into mice that have no immune system. So now if you put them in the kidney capsule of mice, that's pretty simple. You make a flank incision in the kidney, in the mouse, pull out the kidney, use a catheter to squirt some islets under the kidney capsule, withdraw the capsule, and now you have human islets sitting under the capsule of the kidney. And they'll live in this immunodeficient mouse for the lifespan of the mouse, as long as the mouse doesn't get infected with something. So now you can do long-term in vivo studies with honest-to-goodness real human islets. And then you can put in these little mini pumps behind the shoulder blades, and they pump continuously for a month. And you can put whatever you want in them. Obviously, we use harmine and exenatide in combinations. So you can pump them in, you can then harvest the kidney, and then you can clarify the kidney and stain it for insulin. This is more than you want to know, but I'm just getting you ready for the next picture. So what that allows you to do now is to see the numbers of beta cells in a human beta cells in a mouse kidney. So this is a video that Sarah and Adolfo did. And what you can see is, obviously, this is the kidney. Under the kidney capsule, there are these human islets, and they're stained for green. The beta cells are, so this is beta cell mass. So you get a few things from this. One is you get pretty videos to show. But more importantly, since it's 3D, it's voxels, and you're actually measuring human beta cell mass. So now for the first time, you're actually able to measure the mass of human beta cells in a living system or a system that was living until you harvested the kidney. So this is transformative, and it allowed us to do now longer-term studies. So one is, let's take 150 human islets from a donor, put them in the kidney capsule of a mouse, and treat it with vehicle. Or let's take 150 islets from the same donor, but now treat this one with harmine and exenatide, and then we'll measure beta cell mass. And so this is what these kinds of experiments look like. You don't have to have any experience in 3D imaging to see that there's a big increase in beta cell mass in the animals that get harmine and exenatide. You can repeat this experiment a bunch of times. And if you put human islets in a mouse kidney right now and then harvest them an hour later, you have the same number of islets you started with. So the system is actually reliable, sensitive, and reproducible. Since when you transplant them, they're ischemic and have no blood supply, a lot of them die in the first really 24 hours later. So if you come back a week later, there aren't so many, there's about 50% reduction in beta cells. And then if you treat them with just vehicle, nothing, or with exenatide, beta cell mass doesn't change over 3 months. If you add harmine, now you've got about a 3-fold increase in beta cell mass. It doesn't look like much on this scale, but if you think about it, 3-fold is enough to fix most people with type 2 diabetes. And then if you treat with harmine plus exenatide, there's a 7-fold increase in beta cell mass. So this is really extraordinary. This means there are now combinations of drugs that are able to dramatically increase human beta cell mass. Part of this is proliferation. Part of it is enhanced function of the islets. Part of it is, this isn't really a question mark anymore, turning other cells in the islets into beta cells, and really combinations of all of these things. So if you take these numbers, 700% and 300%, and say, if I'm a type 2 diabetic and you tripled my beta cell mass, I'd be up here. If you increased it 7-fold, I'd be up here somewhere. This is like too good for type 2 diabetes. You'd have to use shorter treatments or lower doses. If you look at people with type 1 diabetes and you're starting them after 50 years of diabetes, maybe you can take them from here to here over 3 months. That would be helpful. More basal insulin, that's why we use long-acting insulins. The easier it is to control blood sugar. But if you're a new onset, and we again, as I say, we don't really know the beta cell mass in new onset people with type 1, maybe it's around, say, let's call it 0.3 or something. A 3-fold increase would fix them, a 7-fold increase again would fix them. So this has tremendous promise for fixing both type 1 and type 2. So I showed you that we can make beta cells replicate, beta cell mass increase, but I didn't tell you if they work or not. So I'm going to skip this one because this is really a better slide for a clinical audience. So what we can do is, in this mouse model, we can take these mice, treat them with streptozotocin, and that gives them diabetes. So we get them from the animal facility. They've been eating all night, so their fasting sugar is about 140 in a mouse. Postprandial in the morning is that for mice, and for you and I, it's around 90 or 100 or 80. So if we give them streptozotocin and kill their beta cells, they become diabetic. If we then put in a tiny number of human islets, again, 150 islets, that's not enough to fix them. And if that's all we do and then treat them with just vehicle, they stay diabetic for 3 months. If we give them exenatide, that's a GLP-1 receptor agonist, because of its incretin effect, they make more insulin and their glucose comes down, but it doesn't normalize. If we use harmine in a full dose, it'll completely fix them. So here we didn't want to do that. We wanted to use a low dose. And with harmine alone, beta cell glycemia came down to near normal, but not normal. So the remarkable thing here is that with a low dose of harmine and conventional doses of exenatide, the animals come down rapidly. They stay normal, euglycemic for the 3 months of the experiment. At the end of this study, we also measured beta cell mass. So if you harvest the kidneys here and measure beta cell mass, it's 7-fold higher here than it is in the controls. So you see this 7-fold increase in beta cell mass in honest to goodness, real diabetes. These islets are living in diabetes. And secondly, the beta cell mass increases. It's 7-fold here as it was in the normal mice. But the most remarkable things about this are 2. One is their blood sugar comes down immediately. The beta cell mass hasn't changed much at the end of a week or 2 weeks. So already their glycemia is better. That's telling you that the harmine plus or minus GLP-1 is enhancing beta cell function, beta cell differentiation, independent of its beneficial effects on beta cell mass. And the other part is they have a 7-fold increase in beta cell mass compared to these guys. and yet they don't get hypoglycemic. If we actually inject them with insulin to make them hypoglycemic, they recover rapidly, like normal mice. So you can have it both ways. You can have beta cells mass that expands normally, but they're normally functioning beta cells. They have the good sense to make the right amount of insulin to correct diabetes, but not to overdo it, to turn off insulin when they're fasting or if their blood sugar drops. And then you can also measure human insulin in these guys. And so human insulin levels in these animals are about 4 times higher than they are in the other groups. Okay. So what about safety? So obviously we want to turn this into a therapy that people can use. The enzyme DYRK1A is ubiquitous. Every cell type makes it. So we were worried that we might make other cells in the body proliferate. So in these same experiments, we harvested liver, spleen, intestine, fat, lung, heart, kidney, pancreas, and also brain, not shown in here because there's a bunch of different brain regions we looked at. But the bottom line is there isn't any proliferation in any other organ, just beta cells. We also looked for evidence of neoplasia, worried that this might be driving proliferation in other tissues. And without going into detail, and this stuff is all just published about a week ago, there's no histology, no abnormal histology in any other organ. So it doesn't appear to have oncogenic effects in other organs, and for that matter, in islets either. So what about other adverse effects? It turns out that harmine is a natural molecule that is present in the bark of trees in the Amazon. And you can use those trees or those vines and leaves from other plants to generate something called ayahuasca, which I'm guessing all of you have heard about. If you haven't, you can read about it in the New Yorker or just on Google. So ayahuasca is made from the bark of this vine called Banisteriopsis caapi, and the leaves of this plant called Psychotria viridis. So what people realized hundreds of years ago, and if we have time and questions, I'll say more about this, but people have been doing this for more than 1,000 years. So if you take this vine and these leaves, put them in a pot and stir them, you have ayahuasca. It turns out that Banisteriopsis contains harmine, harmaline, and other harmine related molecules. And harmine and these other molecules are all MAO inhibitors. Psychotria contains dimethyltryptamine. So dimethyltryptamine is a street drug, recreational drug. You can only use it by shooting it up or snorting it. If you take it by mouth, nothing happens. That's because it's degraded in the intestine and first pass in the liver by monoamine oxidase. So what Mesoamericans figured out more than 1,000 years ago is that if you add an MAO inhibitor to oral DMT, now you get the psychoactive beneficial effects of ayahuasca. Okay, so you can, I never heard of ayahuasca before we stumbled on harmine, but I started getting emails from everybody saying, hey, can you send me some, I want to take my family and my kids to Brazil, to the jungle, and have an ayahuasca family retreat. So I started Googling it. And so you can, if you just Google ayahuasca tourism, you can find pictures like this. You can find people who will take you through the jungle, help you identify the plants. You can find a shaman who will chop it up for you, the vines, and put them in a pot for you. You can bring your own shaman from San Francisco if you prefer that to using the indigenous ones. You can throw the leaves of the vines in a pot, cook them up, and then you can just pour them through fine mesh and turn it into this ayahuasca infusion. And then you can have a private party by yourself, or you can bring all your friends and have a group party. This is sort of funny, but it's also real. I mean, if you look, I just pulled all these things off of Google. So the point is, it's widely used in lots of parts of the world, including predominantly Brazil. In fact, in Brazil, there's a religion called the União de Hitau that is associated with ayahuasca use. And so this guy Callaway went to Brazil, found people who do this. They went to Manaus. It says Manaus in here someplace or other. Manaus is where the famous opera house is in the Amazon. Here it is. Here's Manaus. And he found the minister or priest, or they call them master or maitre, in a church and said, "Look, I'd like to enroll 14 of your subjects in your church, and I'd like to do pharmacokinetic studies on them. So can you share with me some ayahuasca so I can measure what's in it? And then I want to do studies on the people when they get it." So the 14 people weighed about 70 kilos, just like us. He measured the amount of dimethyltryptamine in the ayahuasca, 35 milligrams. He measured the amount of, this is per dose, the amount of harmine in the ayahuasca, 250 milligrams. And so they consumed the ayahuasca, and then he did pharmacokinetics. He measured collected blood samples for harmine. So this is harmine here, peaks in around 30 minutes. It lasts for an hour or two and then goes down. This is a degradation product, tetrahydroharmine, so that accumulates as harmine degrades. And then this is dimethyltryptamine. It's lower because the dose of dimethyltryptamine was a lot lower. So this is the time course of harmine distribution in human blood after oral ingestion and dimethyltryptamine. So the peak level that people are getting when they take ayahuasca is around 90 nanograms per mil. In our mouse studies, where we can completely reverse diabetes and increase beta cell mass, the steady state concentrations of harmine are 5 nanograms per mil. So for us, in terms of drug development, this is really remarkable. It means that full therapeutic levels of harmine are about 20 times lower than those that are routinely used in ayahuasca users. So ayahuasca is really used all over the world at this point. People are importing Banisteriopsis from Brazil or from other parts of the world to Europe, to various parts of the U.S. It's actually legal. It's not a controlled drug, ayahuasca, but because this religion, União de Vegetal, also has clergies in the U.S. They went to the Supreme Court, and the Supreme Court said, okay, if you're a member of this religion, you can use ayahuasca in the U.S. Otherwise, it's restricted. Okay. So that's ayahuasca. Ayahuasca is a mix of a bunch of plant extracts made in the jungle by who knows who and what version of plants. So what about pure harmine? Has it ever been used in humans? Well, it turns out it was made by Merck in the 1920s. So this is from the Merck Museum in Darmstadt, Germany. And basically in the 1920s it was made and it was used. It was used primarily for Parkinson's disease and for tardive dyskinesia related to phenothiazines. Turns out it was available, it was safe, but it didn't really work very well. And when L-Dopa came around, it replaced harmine as it and dopaminergic drugs replaced harmine. And so harmine sort of disappeared from pharmaceutical industry in the 50s. So we said it's about time we did a real phase 1 study with harmine and find out if it has hallucinatory issues. That's obviously what we were interested in, or concerned about. So we basically worked together with James Moreau and Jessica Abels here in psychiatry, who are experts in clinical trials and psychoactive drugs. We had some GMP, that means pharma grade, good manufacturing practice, harmine synthesized for us at great expense. But the point is, it's pure pharmaceutical grade harmine. We had approval from the FDA to do this. We had approval from the IRB. We got a grant to do this from NIH. And this is registered in the clinicaltrials.gov registration site, which all drug trials must be registered in. So this is the harmine. It was in 100 milligram tablets. We did this actually in the Clinical Research Center here. And this is the first vial. So it was being dispensed by the pharmacy. So this is a Sinai pharmacy bottle with 100 milligrams of harmine. It was a phase 1 study, which means you're trying to do progressive dose escalations and hit whatever badness you're going to run into. You don't really know what you're going to run into. We were specifically worried that people might have hallucinatory experiences like people have with ayahuasca. So we did a dose escalation. The first subject got 100, then 200, then 300, et cetera, et cetera. So this is actually finished. It's in press. I can't talk about the details of it much for embargo reasons since this is being recorded. But the bottom line is the main adverse event at high doses was nausea and vomiting. So that's interesting because ayahuasca has two adverse side effects. One is hallucinatory, apparently fantastic visual hallucinations, as well as a sort of a mind reset. But also it's associated with nausea and vomiting. And part of this religion is you go to church every Sunday, you take a big dose of ayahuasca, and you have a hallucination followed by severe nausea, vomiting, diarrhea. And so you're cleansed in your brain and the rest of you. So we didn't know whether nausea and vomiting would be part of ayahuasca, and it is. It is of harmine. It is a side effect of high doses of harmine. So this is pretty neat, right? It's going all the way from basic discovery into clinical trials in people. And so it's nice proof of principle that pure harmine is safe and well tolerated in people. But since it's a natural product, you can't patent it. So so if you can't patent it, no drug company is going to spend $3 billion trying to take it through clinical trials. So fortunately, at Sinai, there's a guy named Bob DeVita, who's in the Drug Discovery Institute. He used to be the head of medicinal chemistry at Merck, so he has a lot of experience in drug development. And he has a brother who had type 1 diabetes, so he also got interested in this. And so over the years, together with Bob and Kunal Kumar and Shalata Subaswang, they've synthesized hundreds of versions of DYRK1A inhibitors that are all new intellectual property. They're very potent. Some of them, a lot of them are terrible, right? I mean, they're useless, but several of them are much better than harmine in terms of potency, in terms of specificity for DYRK1A. And most importantly, Sinai now is able to file patents on these. All right. So where we're going, obviously, is to take these next generation DYRK1A inhibitors into people with type 1 diabetes and type 2 diabetes. In type 1 diabetes, what about autoimmunity? If I give you a drug that gives you more beta cells than the same person whose immune system has been destroying them, is that going to work? Well, the answer is probably nobody knows until you try it, but my guess is probably it won't do much unless you do something to protect the beta cells from autoimmunity. A lot is going on here. So you guys know about teplizumab, anti-CD3, that's already approved. There's some others in the pipeline and others not being shown here. But so it's not prime time yet for fantastic immunomodulatory therapy for autoimmunity, but progress is being made in that area as well. So I'm going to leave you with a few predictions. One is that human beta cell regenerative drug therapy is definitely possible. And again, this is a Sinai story. It'll be effective in tens of millions of people with type 2 diabetes initially, and then it'll move to people with type 1 diabetes when immunological challenges are met. It'll be much cheaper and far more scalable than beta cell replacement strategies. I mean, just think of the cost of stem cell therapy and think of the costs of organ transplant, islet transplant, et cetera. So those are going to be expensive. And it'll be at least as safe as other replacement therapies in current use. So for example, pancreas transplant, islet transplant, stem cell therapies are not risk-free. They have their own issues. So what are we trying to do right now? We have this group of more potent, selective, next-gen DYRK inhibitors, and we're moving them through the development. That means you have to, if you want to take them to people, you have to do all kinds of structural metabolism, pharmacokinetic studies in rodents and some other large animal. And then you have to do 14 and 28-day safety studies in those animals before you can do studies in humans. If you think back to the slide where, if you want to take these to humans with diabetes, the question is, how long is it going to take for the diabetes to go away? And that's how you're going to design your trial. So probably, I showed you that it goes away actually in a matter of days with harmine plus or minus GLP-1. So probably we want to do a trial that lasted 28 days or something. That means if you want to do 28-day studies in people, you're going to have to do 28-day studies in rats and dogs. So those are the kinds of things that we're going through now. So that's the long-term large animal preclinical safety studies. If there are adverse effects in tissues outside the beta cell, then we have to come up with a way to target it specifically to beta cells. So far, we haven't seen that, so we don't think this is necessary. And then in type 1 diabetes, autoimmunity, we're not doing anything to make autoimmunity go away. So we presume in type 1, if we make new beta cells for people, they'll be attacked by the immune system as well. So what I love about this story from a medical grounds point of view, I mean, we still have some time, so I'll go off course a little bit. I started out as an internist. I went to med school here at Columbia. I was a resident at Roosevelt. Roosevelt was a great place if you wanted to be a GP generalist. So, and I did, and I was. I finished that, and then I went to. I actually was a GP for a while. And then I decided to do an endocrine fellowship because it was interesting, but I had no basic science training and no particular interest. So I ended up, so I had sort of the bedside package. As a fellow, I learned all kinds of basic science stuff because that's what you had to do in those days. You couldn't say no. I can't say I was dying to do it and I had no experience in it. But I did, and it was fun. So now looking back, okay, I took care of patients with diabetes for about 40 years. I have a pretty good idea what the issues are there. What do they need? It's pretty obvious they need more beta cells. So let's try and work out ways to do that. And then now where we are is we're taking, bedside experience, physician, real life bedside experience to basic science and then back to the bedside. So we're in phase 1 studies now. And you can be sure, like every other drug, it's going to go right back to the bedside. So what I love about this whole experience is the great Mandela turns, and there's room for everybody in every part of this. So in closing, let me just say, this is what I'm telling you is the work of a lot of people. I mentioned Peng Wang, I mentioned Adolfo Garcia Ocaña, Courtney, Sarah Stanley, Bob DeVita. There are lots of others who've helped us with this, including the dean, by the way. I got to say the dean, anything that works on the brain, the dean is interested in. So let me stop there and let's see what we have for questions.
Exciting studies, Andy. And do you envision in your new studies to further document the side effects and efficacy of the harmine analog and the GLP-1 that they have to be given every day for the 28 days? And the other thing I wanted to ask you about was, do you think there's any use for these harmine analogs in Parkinson's disease?
Yeah, so the Parkinson's, I mean, I think the answer to that is no, we have better drugs in Parkinson's, so I don't think it's going to go anywhere in Parkinson's disease. In terms of, Rami specifically, what was the question you asked about the clinical trials?
The dosing.
Yeah, so.
Once a day,
yeah.
Several times a day.
Super important, and so we're actually working through that right now. There's in the studies we did initially, we were giving it just as in mice as a single intraperitoneal injection, so it can be given IP with super rapid pharmacokinetics. But in these studies, like in this slide that I have up here, or let me go back to this one. These were given continuously in these mini pumps, so total flat dose response curves over 3 months. So it doesn't seem to matter how you give it. That has huge implications for us in terms of how it needs to be formulated, and actually we're thinking through that a lot. Maybe it should be given just as one bolus every day. Maybe it should be given 3 times a day. We don't know that, and we're probably not going to know that until we start doing clinical trials. And so that has implications for clinical trial design. Maybe we make a sustained release form and a rapidly absorbed form. In a test tube, it doesn't seem to matter if you put it in, as long as it's in there in the first day. Beta cells are replicating 3 days later. So that has huge implications for drug design. In terms of survival, in terms of, let me go back to this slide. In these kinds of slides, we did, and we as Adolfo, in this case, Adolfo Garcia Ocaña, did an experiment where he put the islets in, treated them with all these conditions, and then stopped treatment, but didn't sacrifice the animal's collected kidneys. He waited another month. And what happened a month later, and that's in this paper that just came out, is that beta cell proliferation went back to nothing. So he stopped the drug, the beta cell stopped proliferating, but beta cell mass stayed up for at least a month. So we can see scenarios where you do this for 3 months and stop it and wait. Those, I think, are going to have to be human studies as well. How long will the diabetes stay away? Et cetera, et cetera. So those are questions that are right on our mind now. We're talking to.
You have to come back to the US.
Yeah. All right. Yeah. Go ahead. What else?
I think there's an online question.
Oh.
Do you think that stem cells converted to beta cells will have much more enhanced response to combination therapy of harmine and GLP-1 agonists?
Yeah, it's a great question. The answer is, so again, just for everybody else, so we have stem cells, but there's cost to producing stem cells. Maybe you could give fewer stem cells and use harmine to help differentiate them and expand them. We actually are doing something just like that in collaboration with the Juvenile Diabetes Research Foundation right now. So the answer is it's a great idea and we should do it, but it hasn't been done yet.
Questions?
All right.
Thank you.
All right. Thanks, everybody.